Thank you so much. I will introduce myself. My name is Miguel Brito, and thank you for the introduction. I'm from the Health and Technology Research Center in the Instituto Politécnico Lisboa, but also I'll collaborate for about 10 years with the Centro de Investigação em Saúde in Angola. It's a research center in Angola where the part of this work has been done, and the other part has been done in Lisbon. And also, it's a pleasure to be here in this symposium, and thanks again the organization for inviting me to participate, because I think it's so important. And in the behalf of my research team, that includes PhD students and other researchers, I will present a project that we are trying to develop in Angola, studying the gut microbiome and sickle cell disease. After several diseases that we have talked today, I will speak about one genetic disease, that is sickle cell disease. And I will not explain in precise the sickle cell disease, but it's a genetic disease, autosomic recessive disorder that changes one of the amino acids in hemoglobin. And in that sense, in cases of reduction of oxygen, hemoglobin will polymerize and will change the structure of the erythrocyte, and it will create the sickling phenomenon that will create several symptoms of the disease, namely vaso-occlusion that can conduct to the organ damage, and also that can conduct to strokes and other pain crises. And also there is a consequence of hemolysis and of course of anemia reduction. So there is a multifactorial diseases that in some part is influenced by the capacity of the erythrocytes to adhere to each other and to create some kind of inflammatory pattern in the disease. In that sense, recent studies performed in sickle cell disease patients. The number of studies is very small for now, and with very limited sample sizes. And they have observed, and people are observed some changes in the gut of the sickle cell disease patients, namely injury in the erythrocyte, changes in microbial composition, increase of permeability. And as I explained in the previous slide, in the case of sickle cell disease, there is some kind of alteration of the inflammation. There is an increase of cell adhesion and age neutrophils that are associated with the vaso-occlusive crisis. So there is a relation between the two conditions, as you can see in this small figure. That this is some kind of rationale of the study. If we have some kind of intestinal dysbiosis, or at least changes in some of the constituents of the gut microbiome, we will have alterations in gut permeability, in bacterial translocation. We will activate neutrophils, we will increase the vaso-occlusive crisis, and this is a non-ending cycle. So in that sense, the aim of our study right now, it's a larger study. I will just present part of the study. The aim of our study is trying to understand in which way the gut microbiome in sickle cell disease patients, in this case in children, sickle cell disease patients, in which way the microbiome can influence the severity of the disease, because there is a great heterogeneity in the severity of the disease, in the phenotype of the disease, and also in which way that the actual treatments can modulate the gut microbiome in order to reduce the symptoms of the disease, and who knows in the future that we can use gut microbiome as a target for therapeutics in order to reduce side effects. In that sense, as I told you, we are collaborating with Angola for about 10 years. Sickle cell disease is highly prevalent in Angola, and we are following a cohort of sickle cell disease patients. And then we select a subsample of about 32 sickle cell disease children that accepted to participate. And then we collect a stool sample from those kids. But in order to compare to a healthy population, we decided to select the siblings of these children that aren't sickle cell disease patients. Mainly they are carriers of the disease, AS, or a genotype. So we choose siblings because they are subject to the same environment, to the same kind of food, the same kind of house, the same kind of village. So in that way, we can reduce the variability of the gut microbiome. Also, in the big project, we are also analyzing some inflammatory markers, television markers. We will not present these results today, that we are trying to use to associate the rationale, as I mentioned earlier. Also, this is a cohort of sickle cell disease patients. They are being followed with treatment with hydroxyurea, and we are also testing the influence of hydroxyurea in changes in gut microbiome. Those results, we haven't been finished the treatment, so I will not present them today. Today, I will just present the comparison between the sickle cell disease patients and the siblings. We use them as a healthy control without disease. And for that, we collected stool samples in the field. They have been protected in DNA chilled fecal collection tubes in order not to destroy the microbiome. They were transported to Lisbon, to our lab in Lisbon, and there the DNA was extracted. We used primers for the V3, V4 hypervariable region, for amplified the 16S RNA region of the bacteria, and then we have prepared libraries to be sequencing by the next generation sequencing in next tech GloMiNe equipment. In that way, after that, we have collected the information and analyzed the count, the number of species, the genera and phyla of that samples. I will present you in a summary the results that we have obtained. And as in some way as expected, and the nutrition is more prevalent in the sickle cell disease children than in the siblings. And interestingly, also we have observed overweight in the siblings, no at all overweight in the sickle cell disease patients, since the nutrition states, as Pascal has already presented, can of course influence the constitution of the gut microbiome. This is a high picture. I will not explain all the picture, of course, but with this graphic, we can, in a way, see that there are indeed very abundant and very conservative groups of bacteria in both of the sickle cell children and the siblings, and you can see they are matched together, the brother in front of the sickle cell disease patient. And in that slide, we can see that indeed there is a difference in one of the genera, the Glaucia. And you can see also the very interesting, because in this slide, we can see in some way the importance of the environment, because there is indeed very similarities between the brothers in a general way, and also variability between the children. Now, what concerns specifically the sickle cell disease patients and the siblings, we observed a very important difference in the Glaucia genus, with a decrease of these bacteria in sickle cell disease patients comparing to the siblings. And also there are other works that I will refer later that associated these species or these genera with a protection for inflammatory diseases. Also, in the sickle cell disease patients, the fetal hemoglobin content is a very important marker of severity, because if we have lower values of fetal hemoglobin, the disease has a worse prognostic, and of course the more severe phenotype. And we indeed observe differences between the most important groups in children with low fetal hemoglobin comparing with children with high fetal hemoglobin. So we have an association between severity and the presence of, or the absence, or the reduction of absence, of course, of several bacteria groups. Moreover, other differences were observed, and with significant differences in the case of Actinobacteria, in the case of the class Actinobacteria, also in the orders. There are differences between sickle cell disease patients. Also in some families, and also in same genders. I will just present a small one because I don't have time for everything concerning the Clostridium group that is increased in sickle cell disease patients comparing to the siblings. And in conclusion, we haven't observed as expected, not as expected, as we expected in the beginning of the study, a difference between the alpha diversity, namely the Shannon index. There is a slight differences between two groups with a reduction of alpha diversity in sickle cell disease patients, but it is not a significant difference. So there is. But however, we have a significant and important differences in several groups, namely Clostridium, as I told you, the group 11, that is a pathogenic bacteria is associated with intestinal dysbiosis. We are further exploring these results, trying to identify the species and which species are present in the sickle cell disease patients comparing to the healthy siblings. We have observed a lower abundance of Glaucia in sickle cell disease children. This bacteria has an impact on health, and it was associated the presence of these bacteria with the alleviating of metabolic and inflammatory disease, which is in accordance to our rationale for this study. And also Romnicox abundance was higher in group of patients and with higher level of fetal hemoglobin. So there is indeed association when we compare groups, sickle cell disease patients with the siblings, and also when we compare inside the groups, it seems that there is a difference in the composition. In the future, no, we are already analyzing those results. We are trying to see if when we increase the fetal hemoglobin with hydroxyurea, if there is some changes also in the microbiome. This is the preliminary result, an important study. There's so much to do, and we think that probably in the future we can think in using gut microbiome as a possible target for treatment of this disease. So once again, I would like to thank all the team, mainly and specifically, I would like to thank Mariana Delgadinho, this is a PhD student. The majority of the work was done by her. Also, Brigida Santos, a pediatrician in Angola that is responsible for following all these kids. It is just a subsample of all the kids and all the institutions, and also the financial support from FCT, and also not specifically in this study, but also from Fundação Gulbenkian in supporting the studies that we are performing in Angola with the support of Portuguese Corporation and Fundação Gulbenkian. So thank you, and I'm opening to questions.
Thanks a lot to Miguel for the presentation. Are there questions already? And then please ask them. Else, I have one actually to start as well. So you showed like really interesting signatures that are associated with sickle cell disease. So do you know, are these the same in Angola as in other countries? Is there like, if you want conserved microbiome change that is associated with sickle cell disease?
There is more 2 or 3 studies, and they have also found some differences. We haven't found yet the same difference they have found, but of course one of the studies was performed in the United States, so the food and the environment is completely different. So now we are trying to analyze and trying to understand. There is differences, of course, but not really the same species, but we were not expected to find the specific species that have been responsible for the differences.
Thank you. Perfect. I think then we go on with the.
Okay, thank you.